Good morning and welcome to another episode of the Atherosclerosis podcast, the podcast series hosted by the EES Young Fellows. So my name is Sanne Maas. I'm from the RWTH Aachen in Germany, and I will be your host for the episode of today. Today's guest is not only advancing our understanding in dyslipidemia, but also brings the science to the clinic. And we're thrilled to welcome Dr. Simon Pfisterer, who is the group leader of the Pfisterer Laboratory at the University of Helsinki and the co-founder and chief scientific officer of a startup company. His research group combines laboratory automation with cell biology, elucidating how genetic variants contribute to biological variability and how biological variability links to disease progression and treatment outcomes in dyslipidemia and cardiovascular diseases. And with this approach, he aims to disentangle genetic complexity of dyslipidemia and develop novel tools to improve the diagnosis and treatment of familial hypercholesterolemia. So, hello, Dr. Pfisterer, and welcome. So it's a pleasure to have you today and to talk about your career, research, and your perspective and the future of atherosclerosis research. And on behalf of the EES Young Fellows, I would like to thank you for accepting our invitation for this interview during the 93rd EES Congress in Glasgow in the UK.
Thanks. I mean, it's really great to be here. Thanks. That was a really long introduction.
Yes.
So I'm happy to be here, and I'm Simon. So
yes.
Yeah.
I can call you Simon during this interview.
Yes.
Great. That's very nice. So in the series of interviews, we want to give our listeners a chance to get to know you a bit more. Both personally, also research and academic career level. So could you please introduce yourself to our listeners?
Yes. So I'm Simon. So I'm a biochemist. Studied biochemistry in Germany, and then focused really on cell biological research, doing research not only in Germany, but also US, Australia, and now in Finland. And the main focus was in the beginning, especially PhD and postdoc research on really basic research, cell biological concepts of, for example, autophagy and intracellular recycling system, but also intracellular cholesterol transport then during my postdoc, cholesterol transport at membrane contact sites within cells, and also lipid storage within cells. And then in 2019, I established my own research group where the main goal was to combine laboratory automation with this cell biological research, because I always felt like cell biology is great, but it's like so slow and not very systematic, and the results can be varying a whole lot. And I really felt like we can do a change by integrating laboratory automation, trying to get more systematic insight, more results over time, and really hoping to make a change in kind of the discovery making in this process.
Yes, that sounds like a good tool that we need indeed. Yes. So thank you for this answer. Could you maybe also describe how your typical day looks like, if you have such a day, and how you then also balance your different responsibilities that you have as a researcher, but also with the company and being a co-founder and also being a CSO?
Yeah, maybe I'll start going like a little bit backwards. So when we established the research group, we were focusing on these two kind of key aspects. What's a functional consequence of a genetic variant, which I think is a big problem because we know a huge amount of genetic variants, but we don't really know how they affect cellular disease mechanisms. So that was one aspect that we are working on in the research group. And the other aspect was that we wanted to look into the same cellular disease mechanisms, but how are they different between individuals? And this, in the end, we were then getting commercial funding in a way from Business Finland to acquire additional experimental evidence, but also to have funding for patenting and so on. And then we moved this into a spin-out company, which was established last year, which is Moncyte Health. So now my lab looks a bit, or like my day routine looks a bit scattered in a way. So usually I start in the morning, I start in the offices of the company with meetings and discussions. ongoing there. And usually in the afternoon, I move to the research group side. Luckily, both of them are, like, of course, very well connected. It's a spin-out company from the University of Helsinki. We still share the same lab space, cell culture facilities, microscopy equipment. So there is lots of overlap. And I think that's, in a way, the nice part, in a way, of it.
Yeah, makes it easier for you to combine both roles.
Yes, makes it easier to combine both roles and stay in touch really with the development on both sides.
Yeah. So you already gave a bit like an answer partly to the next question. Do you have any advice for those aiming to starting and to manage multiple responsibilities at the same time?
Well, I think maybe first of all, I feel like it's like extremely rewarding to have multiple responsibilities. And I think really trying to translate the basic research into a biomedical application has been like a really great experience. And it opened up lots of opportunities. And you think you get lots of different experiences along the way, from talking to clinicians and other stakeholders on can we translate the technology, but also all of the other aspects like quality control, regulatory aspects that come along. So I think this is like always then a feedback cycle. In a way, it's enriching also the research group, the perspectives in other areas. And I think, therefore, it's like super rewarding to have these multiple roles and responsibilities. Of course, it can be challenging to juggle them in a way, but I think the most important thing is that I have excellent people. I'm not doing this alone. So in the research group, or in the company, I have a co-founder, Tamara Alagirova, who is the CEO for the company, who's handling a lot of the commercial aspects, investor relations, and so on and so forth. We have excellent members in the research team of the company, but also in the research group. So I don't need to do everything myself.
That's good. Yeah. Yeah, we heard this already before, that delegating tasks is very important if you have so many responsibilities. Yeah. So we got to know you already a little bit better. And we would like to also talk with you about the research, of course. So could you tell us a bit more about your academic journey and what led you to study and focus on dyslipidemia, cardiovascular disease, and also particularly the familial hypercholesterolemia?
Yeah, sure. I mean, what I said before, I'm a real cell biologist. So I started with if we think about membrane contact sites, like how is cholesterol transported between membrane contact sites? I did a lot of microscopy checking like how cholesterol is moving within the cell. And also one key aspect was about lipid storage, like how is fat, lipids stored within lipid droplets, how dynamic are lipid droplets, and so on and so forth. And that was a lot of like live cell microscopy to visualize these aspects within cells. And of course, that is related in a way to the real disease mechanisms in dyslipidemia, cardiovascular disease. So that's where it started then from. I really felt like for establishing my own research group, I really want to make things different to what we are doing before. So therefore the move towards automation. So now we are not going to the microscope anymore and select representative cells. So we have a fully automated microscope that takes the pictures. We have robotic tools to prepare the cells for microscopy. We have software tools to automatically quantify each individual cell and to get the data out. And then, of course, we get a lot of data. So we need to do Python scripting, programming to analyze all of the data that comes out of that. And this is like how I transitioned in a way from this like really basic research towards, how should I say, more an I would not necessarily say, for example, the genetic variants, of course it's applied. It has enormous consequences. But this shift towards automation, of course, gives us the unique opportunity now to look at a lot of gene variants. Like beforehand, gene variants, LDL receptor variants have been characterized throughout the last 30, 40 years. But it has been always like 10 variants, 5 variants that have been characterized and where the results have been reported in a manuscript. And now we have the technology, and I think this spring we published a paper where we have been reporting the results of 315 LDL receptor variants, and where we could link this result then with whole exome sequencing data from UK Biobank. So this gives a lot of opportunities, and I think this really, I feel like it can make a change in, for example, diagnosis of familial hypercholesterolemia, where genetic tests are actually part of the process, but in a lot of situations, it might be that a doctor gets a result where it says, this is a variant of unknown significance. So what do we do then? What do we tell a person living with FH in this case? So I really feel like that our research, considering the genetic variants, has a real impact and can have a real impact in the diagnosis. But what we realized is that the genetics of familial hypercholesterolemia, dyslipidemia, is extremely complex. Mm-hmm. And therefore, this other approach that we now moved into the company setting, I feel like is extremely exciting because it allows us to look at those disease mechanisms, cellular LDL uptake, lipid storage, and get the entire integration of this genetic complexity that we have challenges like deciphering, and in combination, potential contributions of lifestyle and other factors that are really influencing our biological processes that relate to disease, dyslipidemia and cardiovascular disease. So this is in a way then how we really ended up transitioning from a real basic science aspect that could, for example, lipid droplet research can be linked with obesity, a lot of other disease applications. We are now much more focused on dyslipidemia and cardiovascular disease. But I still believe, especially in the company setting where we look at cellular disease mechanisms, every disease or nearly every disease is linked to alterations at the cellular level. So I believe that we can translate this technology not only into medical applications in cardiovascular space, but also other disease applications.
Yeah. That's a very interesting answer. And indeed also that you have really these two concepts that you link now together, that you have the basic science and the company that you can really study both. So what was your vision when you decided to co-found the startup? And what have been the biggest challenges and learning moments in translating academic discovery into the company?
Oh, that's a
tough one.
You want the long or the short answer? As a basic researcher, I think, or at least you have an impact session where you always discuss how would this help the general public, where would this have an impact for society, and so on and so forth. And of course, we always say that this has an impact, but when we got the first results from the familial hypercholesterolemia patients, I really felt like, okay, now there is a possibility actually to turn this around and actually maybe translate this into an application and bring it in such a setting that we could really help someone. Mm-hmm. And then I started to realize in a way that if I just publish this, then I think not much will happen. So I really felt like that I need to take some key steps of developing this further. And then this was kind of the stage where I decided to apply for this Business Finland funding, where I got help in a way from the university and where I could get key people on board that really helped like moving this forward, like the now co-founder and CEO Tamara, and other people who really helped along the process. And but then there are lots of different aspects. I think one key aspect was moving the invention, the patent that we filed at the university into the company. We went through long negotiations about the university equity share in the company, potential royalties and other things that need to go, which is in a way a bit like, how should I say, at this stage, we were thinking about making the company and getting demands on equity share, royalties and everything, where you're just developing the technology. You see, you have to put in a lot of effort into bringing the technology into a setting, was quite, in a way, a challenge for us at that stage. And it took really long time to get it there. And I think the other aspects are then really about quality and the regulatory aspects that relate to translating something from a basic research setting, protocols, standard operating procedures, and everything into a more consistent, systematic approach where you have the different checks and balances in a company setting.
Yeah. Would you also have an advice for researchers that are interested to turn their academic discoveries into a successful startup?
Do it. I think that's in a way. I think the key thing why, at least I am doing science is because, I would like to discover something new that is actually helpful in a way, somewhere, in some place. And I feel like that we have a lot of basic research discoveries where there are lots of potential applications, but we are just not taking the next step of bringing it into a real application. And I think, in a way, it's our responsibility as scientists to do this step, to not only publish our findings and hope that someone else will pick it up. Of course, that's working in some cases, some exceptional cases, for example, also drug development or so on. But I think if we really want to bring something novel into into an application, we really have to put a lot of effort into actually making that happen. And it's more like encouraging a lot more people to do it, because I think then we can have a lot more change and have a real impact of science. And it's our responsibility. We get taxpayers funding to run our research and science. It's our responsibility to, in a way, not only publish it, but also translate it into something and try to have a real impact for people in the medical or other settings.
Mm-hmm. Yes, yeah. I think that's a very good point. And during the Congress, you're also giving a talk about the novel therapeutic approaches in atherosclerosis. So can you maybe already give us a glimpse of your talk that you will give, and what will be your take-home message? And what do you hope that the attendees will remember from your talk?
Yeah, of course. So, I will talk about this aspect where we perform like cellular analysis on LDL uptake, lipid storage in the white blood cells that we get from a 10 ml blood sample. And the key thing here is that there's huge biological variability in cellular LDL uptake, maximum LDL uptake capacity between individuals. There are huge differences in lipid storage, in, for example, circulating monocytes, which have been proposed before to play a key role in inflammation, for example, atherosclerosis progression. And I really hope that I can convey a message that it's like really important to look at these aspects and that there is a chance to utilize these technology to get better insight into, for example, residual cardiovascular risk, individuals who are taking statin medication that have a poor cellular profile, have a pro-atherogenic lipoprotein profile, have higher cardiovascular risk, and are not achieving their treatment goals, for example. So these are, I think, really exciting aspects. I think they are really important. I'm convinced that it can help us to advance precision medicine, and I hope that I can convey this message also to the audience in the end.
Sounds good. Yes. So I would like to shift now to the next subject that we have, so the expert perspective. And I would like to ask you about current challenges and innovation in the atherosclerosis research. So how is a multi-parametric platform different from the existing assays?
Yeah, I mean, I would say if we look at the most commonly used, let's say, biomarkers that are there, it's of course like LDL cholesterol, blood lipid panel measurement, maybe NMR metabolomic data, Lp(a) measurements. And in a way, it's a bit heartbreaking to see that we are still fighting to increase the level of Lp(a), for example, testing, and to, in a way, get with these like minimal readouts already like a decent understanding of a certain risk profile. But in the end, it's biomarkers that are a consequence of something that has been going wrong at a cellular level. And the biomarker itself doesn't necessarily give us a good way on how what is the real risk, for example. If we think about LDL cholesterol, the real risk of getting a heart attack might still be very different for individuals with similar LDL cholesterol values. How they respond to certain medication is not given from a blood cholesterol measurement. It's in a way, at this stage, I would say, approached in a trial and error fashion. So our platform, in a way, is providing very different insight. It's really looking at those cellular disease mechanisms. Cellular LDL uptake, lipid storage are key components of the, let's say, cellular drivers of dyslipidemia and in the end also cardiovascular disease. So this enables us really to look into how are they different in individuals, and then we hope we can leverage this technology to optimize treatment selection, but especially to give a better insight into the cardiovascular risk. And I think that's then a really exciting opportunity to have the chance to say, okay, wait a second, this person is at much higher risk for a heart attack or stroke. And at the same time, we can say, okay, we need to be more careful with the treatment selection. We need to be much more aggressive, and we can see that this person, for example, will never respond to statin medication alone and will need much more like combination therapy to get to then the lower targets that are relevant for such a person.
Yeah.
Yes. Did I answer the question?
Yes, you did.
Yes. Yes. So, I mean, that's, of course, like our multi-parametric platform. And I think it's different to a genetic test, very different, I would say, to a genetic test, where the genetic test we can see with, for example, familial hypercholesterolemia patients, heterozygous familial hypercholesterolemia patients. You would expect that the person with a loss of function LDL receptor mutation achieves, for example, 50% reduction in cellular LDL uptake. But we can see that there are enormous differences between individuals with the exact same gene variant, that some individuals have only 20% uptake, whereas others have completely normal LDL uptake. And we can see that this, in a way, links with what these people achieve when they are on treatment, on statin treatment. So yeah. I think that's exciting, and it shows that we don't understand the full complexity of the genetic contributions, and therefore I feel like there's a huge benefit of actually looking at the cellular mechanisms from a person's blood cells.
Yeah. So your answer already links a bit to the next question. So in your group, you're also contributing to the characterization of this LDL receptor variants. So do you see also a future there in genome editing in correcting high impact mutations? And if so, how far are we from clinical implementation, considering current challenges?
Well, yes, that's of course a big jump now. I mean, I think well, first of all, I think it's extremely important to accumulate a lot more data on what's the real consequence of I think before we start necessarily trying to modify the genome, I think it would be good to actually understand whether one of these gene variants is actually having an effect. If it doesn't have an effect, we don't need to modify it.
Yes.
And then I think, like what I tried to say before, like having the real functional data for a gene variant will be extremely helpful. But on the other hand, I still think it's more like a ballpark estimate on where we are going with cardiovascular risk and the other ones. Now, relating to the question of like real gene editing in a clinical setting or as a therapy option, I think, well, I mean, I think there are, very elegant approaches now at this stage where I feel like, for example, trying to modify each individual LDL receptor gene variant is very challenging because you will need to control off-target effects for each of the designs for each of the variants. So I think what we can see now with, for example, targeting PCSK9 or ANGPTL3 is a very elegant approach because you can make one design for your gene editing. You can very well, or try to very well, control off-target effects and thereby circumvent these off-target or checking the off-target effects for, let's say, thousands of different gene edits that you would need to do in, of course, like individual persons. But I think like over time, when we are able to control these aspects of off-target effects and other events much better, I think also it will come to individual like editing of gene variants. But still, I think it would be good to know what is the effect of a gene variant? Does it really have a functional consequence? Can it be adjusted with a simple medication, or do we need gene editing for it? And even for the current approaches with PCSK9 gene editing or ANGPTL3 gene editing, still it's very important to know if there's a person who has absolutely complete lack of cellular LDL uptake. Even if we change PCSK9, it will not necessarily help the person a whole lot. But if we detect a residual LDL uptake capacity in those individuals, of course we can, and then the PCSK9 gene therapy will help.
Yeah.
So I think that's in a way also exciting opportunities for diagnosis and relating it then to the follow-up gene editing.
Yeah, we really need this personalized medicine and look more into what the person itself needs. Yeah. Good. I would like to thank you for the answers that you gave now. Now I would like to go to a bit more general topic. So within this podcast, we also want to inform our listeners about the possible open positions in different academic institutions around the world. So do you typically advertise open positions, or would you encourage interested PhD students or postdoc candidates to reach out to you directly?
Yeah, of course. I mean, if there's someone who is interested, I mean, a direct contact is always better. Of course, I mean, it depends on whether there is funding available, whether you have, in a way, a possibility of recruiting someone. But I think you never know, in which situation you are currently are. And it's always nice to not necessarily needing to make a a public like announcement for it. Even though we, of course, sometimes you just get the funding and then you need to recruit from there. And of course, we are also recruiting very talented, very excellent persons from a general call. But so both things are happening in a way. But I think if there is someone who is really interested in doing something, if it's a student who wants to come over just to, in a way, it's also, I feel like, starting with an internship or something enables, in a way, also for the person joining to actually figure out, is this something that they really feel like it's interesting for them? And I think, in a way, it's research, and I think research should also be fun because we spend a lot of time with it. And if you realize in an internship, okay, this is maybe not something that is really exciting for me, that is not a lot of fun to do, then maybe it's actually a great opportunity also to say, well, then maybe I go somewhere else, I try something else. And I think that's just part of the process of finding something that is really interesting to both sides in a way.
Yeah, yes. So the applications, where can they, like when you have an announcement, do you mainly share this via LinkedIn or also via the website?
I mean, I'm lazy updating the lab, the lab webpage, but of course it's announced on LinkedIn. And then, well, if it's for the research group at the university side, then the university is making a real job advertisement, which is then like distributed. And I think it's ending up on a lot of different job portals in the end. For the company, we have been mostly advertising via LinkedIn.
Okay, yeah. So then people know where to follow you and see if they're interested to work with you together. So it has been a fantastic conversation. Thank you very much for sharing your time, experience, and insights with us. So thank you for this interview.
Yeah, thanks so much. I think this has been great fun. So,
great.
And thanks for having these talks. I think it's really valuable for the community. And yeah, thanks for all of your efforts.
Thank you very much. Yeah, that's also something we would like to share with the audience. And we would like to thank also thank our listeners, because this podcast is for them. We also need their feedback to tailor the interests so that it fits what you need, what you want to hear. So if you enjoyed this episode, please share it with your colleagues and fellow researchers and let us know who you would like to hear from next. So please don't forget to leave us your impressions and suggestions for topics. What would you like to hear? And please stay tuned for the next episode. So thanks again. Have a great ES. And we're looking forward to your talk during the conference.
Thanks, then. Same to you. Enjoy the conference.
Thank you very much.